Today, you'll be inoculating 2 tubes of 1% tryptone, 1 tube of 1% tryptone and 1% glucose, 2 tubes of MRVP test medium, 2 tubes of citrate agar in slants, and lastly, 2 slants of Klinger iron agar for H2S production. I hope each of you enjoyed last week's look at the various microorganisms and their abilities to use enzymes on carbohydrates and proteins. This week's exercises address our ability to identify various biochemical byproducts. These can be detected by adding a pH indicator to the culture medium following incubation, or by adding 1 or 2 reagents and examining the medium for a specific reaction which exhibits a color change in the surrounding medium. Collectively, the Indole, methyl red, Vogt-Pascal, and citrate tests are referred to as the IMVIC series of tests. The IMVIC test is used to differentiate enteric organisms. However, we will be using these tests to aid in our identification of the other microorganisms we'll be testing in this lab. Enteric organisms are gram-negative rods that are typically found in the intestines as part of your normal flora or occurring pathogenically. The first test that we will discuss is the Indole test, which involves a 1% tryptone broth preparation. Indole is a compound formed from the breakdown of tryptophan, which is an amino acid, by the enzyme tryptophanase. By doing so, the organism is able to use this for a variety of cellular processes and produces the chemical indole as a byproduct. We are able to detect indole production by adding a small amount of Kovacs reagent to the medium after incubation for 2 days. When conducting the indole test, you will tip your tube, as indicated, to the side, allowing 2 to 3 drops of the Kovacs reagent to the medium, allowing the Kovacs to slowly form a layer on the surface of your culture medium. Next, you will cap the tube and slowly rotate the tube in your hands. Organisms that are able to break down tryptophan will turn a reddish color after addition of Kovacs, indicating that the indole was in fact produced. To illustrate an important concept today, we will also be testing an organism in a 1% tryptone, 1% glucose broth tube. The purpose of this is to highlight the fact that microorganisms, like us all, prefer to take the easy way out. With this test, you will notice that the organism will prefer to utilize the glucose over the tryptone in the culture medium. This will result in little to no indole production and a brownish color instead of the typical reddish color you get from indole production. The next test is a combination test. MRVP test medium is used to detect an organism's ability to form neutral byproducts, specifically acetamethylcarbinol, or AMC, through their use of this medium. Since this test is basically 2 tests in one, I like to advertise this as a twofer, meaning you get 2 tests for the price of 1 when it comes to your working on your unknowns. This test begins like any other, with an inoculation into the medium, which is then incubated for 2 days. This test should actually be run after 3 days, but due to the nature of this lab, we will check it a day early. After incubation on day 4, you will remove 1 ml of the culture medium from the initial tube, tube A. Place this into a clean test tube labeled tube B. It is important to conduct the right test on the right volume, as the reagent amounts have been adjusted for these specific volumes. To the 1 ml tube, tube B, you'll add the necessary drops of alpha-naphthol and 40% potassium hydroxide, as spelled out in your lab manual, and can be found in dropper bottles on your lab bench. This tube should be left for up to 15 minutes and should periodically be aerated by simply tapping the tube lightly against the side of your palm. Now let's focus our attention back on tube A. To this tube, you'll add the appropriate amount of drops, as stated in your lab manual, of methyl red. Methyl red can be found in dropper bottles where you normally find your immersion oil for microscopy. It is important to know the difference between methyl red and phenol red. Methyl red behaves in the opposite manner that phenol red behaves, and your TAs will discuss this. So a red color found in a VP test 2B will indicate that the organism produces AMC. A positive result, a red color found in the MR test, tube A, indicates the medium is acidic. This result would also be recorded as a positive result. Therefore, if an organism is positive for VP, it will almost never have a positive result for MR, unless you're working with more than one organism. Remember your unknowns? The last test we'll discuss as part of the INVIC series of tests is the citrate test. Sodium citrate is a component in this medium and can only be used by certain microorganisms as a carbon source. Those that have the ability to produce the enzyme citrase can utilize this. By doing so, the organism creates an alkaline or basic environment in the medium, which is detected by the pH indicator bromothymol blue. At an alkaline pH, the medium turns a beautiful blue color. Those organisms that cannot use the citrate as a carbon source will not change the color of the initial medium, and the medium will stay green. That concludes our discussion of the INVIC series of tests. Let's now discuss the remaining two tests, these being the hydrogen sulfide test and the Catalase test. The hydrogen sulfide test can be prepared in two manners. The first is that you can prepare it only as a stab, or the second way is that you can slant the medium. There are more benefits to slanting the medium, which is why we use this method. The name of the medium that we will use is Klinger iron agar. Klinger iron agar contains lactose and dextrose at a 10:1 ratio respectively. In the anaerobic environment of the butt of the tube, organisms that can utilize the thiosulfate in the medium produce H2S, acid, and possibly gas. H2S, when present, reacts with the iron in the medium to form a black precipitate. As you are all aware, sulfur has a very distinct smell, and if you uncap your tube and waft the air toward your nose, you will surely detect this. To inoculate this test, you will need to obtain a loop and a stab. First, you will stab the butt of the tube using the stab. Next, using the loop, you'll obtain the organism and streak from the bottom up to the top of the slant. Streaking the slant will allow you to obtain additional information on your organism. Depending on where along the slant there is a yellow color, will inform you whether the organism is able to ferment lactose or dextrose or both. Your TAs will discuss this with you in detail in lab. Gas can be detected by examining the butt of the tube. Sometimes you may see a few different ways this is exhibited. The medium might be raised, cracked, or split completely depending on the amount of gas produced by the organism. In order to view an accurate result of this test, you should check it after 1 day of incubation. TA should set these tubes up in separate racks for their section so that I can easily remove them from the incubator and refrigerate them. The last test we'll be conducting today is the Catalase test. Organisms that produce the enzyme Catalase are able to break down hydrogen peroxide into its components, water and oxygen. This test is used primarily to differentiate Staph species from Strep species. Bacteria that can use oxygen will produce Catalase. Those organisms that do not produce Catalase will not form oxygen from the addition of hydrogen peroxide. For your unknown, this test will only give you a negative result on one of the organisms. You will see today which organism that is. To conduct this test, aseptically remove the organism from the culture tube along the slant surface. Place the organism on a clean and dry microscope slide and simply place 1 drop of hydrogen peroxide on the organism. Oxygen production will appear as bubbles and can be detected with or without the aid of the microscope. Depending on the amount of organism you place on the microscope slide will determine how much bubbling you may see in the drop of hydrogen peroxide. This concludes the Bacterial Enzymatic Reactions Lab. Just know that these tests are just a fraction of the many tests that a microbiologist has at his or her disposal. However, in this lab, you'll be restricted to just the biochemical tests that are listed on your unknown chart. I hope you have learned a great deal and that you will be well prepared for when you work on your unknowns.